everyone, I am Mansi Andharia. I am from Biomedical Genetics Department of Vellore Institute of Technology. In today's demo lecture, I am going to talk a bit about DNA replication and the process of DNA replication and enzymes involved in it. So we know human beings are made up of organs. Organs are formed by a group of cells called tissues, and these tissues are made up of cells. Cells have nucleus and nucleus have genome. Cells divide and we grow. But have you ever wondered how the genome is getting divided into two cells? We know the processes of mitosis and meiosis, but the cell have to double first itself in the original cell so that we get the same genome in two cells. To explain this, first thing is DNA replication. So DNA replication is a biological process where the double helical structure of DNA gets divided into two identical strands that is also double helix, two identical strands. So we'll just start by a little brushing up about the cell cycle. So it has G1 phase, it has S phase, it has G2 phase, it has M phase. DNA replication happens in the S phase of cell cycle. G1 phase is a growth phase that prepares the cell for, cell grows in its content of nucleus and all. It will make the cell ready, it will have nucleotides and all. It will make the cell ready for the S phase. Later in the M phase, we have the cell division through different mitosis and meiosis processes. Why I brought this over here, I want to say that DNA replication happens in the S phase of this cell cycle, where the double helical structure of DNA gets divided into two identical structures. That is how one becomes two. So this is it. And if we talk about DNA replication, this is DNA. Just basic sentence. It will get divided it will have two strands. One will be similar from the over here. Now how we will see later. We'll see this later. So DNA replication, first enzyme that comes in the picture is helicase. Helicase is an enzyme that has capacity to unwind the DNA. Now, have you ever wondered DNA is such a complex, it is a double helix and it is such a thing that is being in the cell which is so congested. It has to have something in order to hold it in its place, right? What is it? So there is something called as hydrogen bonding, which is very important over here in DNA replication. Why am I bringing it over here is the helicase enzyme that we are talking about, which is the first enzyme that starts the DNA replication, has the capacity to break this hydrogen bond and unwind the DNA. Helicase has the capacity to break the hydrogen bond and unwind the DNA. So how does this unwinding happen? There are two strands, 5' to 3'. One runs from 5' to 3'. Other runs from 3' to 5'. Both of these strands are called as complementary strands. Why? Because if there is A here, there is T here. If there is G here, there is C here. A and T binds with triple bond, G and C binds with double bond. And they have very strong hydrogen bond between them, hydrogen bonding forces. These forces keep them intact in the structure. But helicase, as we talked about, have the capacity to bind to what? It binds not anywhere but to the origin of replication. There are sequences in DNA which have particular nucleotides and particular properties called as origin of replication, and helicase has the property to bind to these ORIC, origin of replication sequences, and unwind the DNA and form something called as replication forks. So it forms two replication forks. After forming two replication forks, there are two strands, both of which are called as template strands. Now if you see, they originate from the original DNA only. These two template strands originate from the original DNA, and the new strands formation happens with the help of these. It acts as a template, it guide. This strand and this strand act as a guide for DNA polymerase to form another strand, right? So there are two replication forks with the help of helicase. binding to the ORC region have formed two replication forks. One is called as now one strand runs from 5 prime to 3 prime, other runs from 3 prime to 5 prime. We know there are two complementary strands in DNA because like one, if there is A here, there is T here, if there is G here, there is C here. A is definitely gonna form triple bond with T, G is definitely gonna double bond with C. Wherever there is A, there has to be T, or wherever there is T, there has to be A. Wherever there is G, cytosine, has to be guanine. So these are the nucleotides, and they have hydrogen bond. Helicase have opened up with the by binding into the origin and formed two replication forks and two template strands. Now, one thing that we need to know is about the hero of replication, that is DNA replication, that is called as next enzyme is DNA polymerase. Two conditions, two very important conditions that DNA polymerase have given to the replication procedure is that should have primer, it runs only from 3 prime to 5 prime direction. It should have primers and it runs only from 3 prime to 5 prime direction in this direction. DNA polymerase runs in, for this template strand if I say, it runs from this 5 prime to 3 prime direction. Now, one strand is 5 prime to 3 prime, other strand is 3 prime to 5 prime. So, how is that going to happen? That happens by Okazaki fragments, which I will talk about it later. But now, let's see what happens after helicase have opened up. There are two strands. Primase is the one that is first thing that forms primer. The first condition of DNA polymerase, primer. Formation of primer happens with the help of primase. So, I told DNA replication, DNA polymerase acts only in 5 prime to 3 prime. So, if this strand is 5 prime to 3 prime, obviously the other has to be from this side, like this, somewhat like this, 5 prime to 3 prime, right? So, primase comes here and forms and adds short strands of nucleotides. Like a short sequence it will form. Like see, initiation of second strand have started already. Similarly, over here, if it is 3 prime, DNA polymerase act only in 5 prime to 3 prime. So, over here, primase will come. It will add short strand of nucleotide. So, what does the enzyme primase do? First enzyme was helicase. Second enzyme is primase. Primase add short nucleotide sequences. Okay, maybe 10 to 12. 10 to 12 RNA sequences will be formed by primer. Now, only if these sequences are formed is when the DNA polymerase can come and act and add nucleotides further and further take ahead the replication. So, after adding these short strand of sequences, this is leading strand, DNA polymerase will come, act and over here. But now, one thing is, this is lagging strand. This strand is from 3 prime to 5 prime. Template strand is 3 prime to 5 prime. So, the primer will form Okazaki fragments. There will be so many short strand of sequences formed before. This is because DNA runs from, because to DNA have two complementary strands. One strand is 5 prime to 3 prime. Okay, DNA polymerase can come and form from here 5 prime direction to 3 prime continuously because it has a property, it adds 5 prime to 3 prime. But the next strand is 3 prime to 5 prime. Complication comes here, and that is like a strategy that our body has formed to form Okazaki fragments, small fragments that DNA will form. So primer also small will be there. After which, after formation of primers, primase is going to leave the case. Now primase is gone and primer is there. So first condition of DNA polymerase to act was primer. Primers are present. So of course DNA polymerase will come in the picture. So this is 5 prime. Over here this is 5 prime because this is 3 prime. Complementary strand definitely have to be 5 prime. So DNA polymerase will come over here. It will come over here. So many DNA polymerases will come. And it will, now as DNA polymerase have come, it will start forming short strands. It will start adding nucleotides from See, the second condition runs, or better than I'll say adds nucleotides. Adds nucleotides only from 5 prime to 3 prime direction. So this is 5 prime, because this is 3 prime, has to be 5 prime, so 5 prime to 3 prime. It starts adding nucleotides. Over here also DNA polymerase, this is DNA polymerase, and it forms over here a continuous strand. Over here continuous, over here lagging. So this strand is called as a leading strand, and this strand is called as, simply because it is not adding in continuous direction, because of this 5 prime 3 prime thing, it's called as a lagging strand. So now DNA polymerase has started adding nucleotides, and see, we can already see there is formation of new strands begin. But here there are fragments, and here we have a continuous strand that is being formed. So what we saw till now is that there is 5 prime to 3 prime template strand after opening, and there is 3 prime to 5 prime, 5 prime to 3 prime. 3 prime is always a leading one. It is called as a leading strand. This is a lagging strand. Okay, both of these act as a template for DNA polymerase and primase to come and act. And now, as this is a leading one, 3 prime, so 5 prime, this is a short primer, if you remember, a primer that is formed by primase. Yeah, 3 prime to 5 prime, lagging, leading, 5 prime to 3 prime. Okay, so what we have seen, these blue color ones are primers. This is the template DNA that was there here already, right? So short primers sequences, not to forget, from 5 prime towards the 3 prime have been started forming. Short primer sequences have formed with the help of primase. DNA polymerase have come, and it has also started adding its nucleotides. As this is leading strand, it's going to add continuously, because it works like this. It works from 5 prime to 3 prime only. And in Okazaki, it is short sequences. Like here there will be a DNA polymerase, here there will be a DNA polymerase, here there will be a DNA polymerase, and it's going to act and add nucleotides. After adding nucleotides, now this is the scenario. What happens next? Primers have to be removed. We do not need RNA primers in our DNA template, right? So removal of primers is the next step. How does this happen? Another enzyme that come in the picture, exonuclease. Exonuclease are the class of enzymes that have capacity to remove particular nucleotides from DNA. They have the capacity to remove particular nucleotides from DNA. So exonuclease is going to come, and it will remove the primers. After the DNA polymerase have gone, it is going to remove the primers that were formed by primase initially, like soon after the helicase have unwind. Those primers are removed now, and as DNA polymerase already have a little template, kind of, not template, but a little sequences over here, it can then again come and complete the formation wherever there is place. Like if we remove primer from here, like 1 fragment, 2 fragment, 3 fragment in lagging strand, and a complete in a leading strand. Now one more thing, we have gaps over here, and there is no gaps in DNA replication. It doesn't work like that. So what happens? Another class of enzyme, ligases. Ligases are the class of enzyme that have the capacity. It is called as sealing enzymes. Seal the DNA. They have the capacity to seal the DNA and join this DNA. Just joining of these breaks. That means joining of. Initially there were here Okazaki fragments, right? Over here there were Okazaki fragments. In the lagging strand there were short fragments that were there. Those are Okazaki fragments. So DNA ligase has the gap. After we remove the primer, whatever gap is there is filled by DNA polymerase, and after gap is filled if there are any kind of, there are spaces between them which is filled by ligases. So ligases seal the DNA and make it like a normal one, or like a leading strand, which do not have any breaks in it. So this is about DNA replication. There are two strands that is formed. Now if we see, this was the template strand. This what we formed is the lagging strand. This is again template, black one. And what we formed over here, we formed a new leading strand. Now, after which this DNA, this can act as a separate, it will undergo the helix structure again. Even this can undergo a separate, and it will be something like this. There was this DNA over here. The new strand that have formed over here, over here also. Now see, this is very clear now. This was the mother DNA. Just calling it as mother DNA, just naming it. Okay. This black from the original DNA is the template strand. And the new one, see there is new DNA formed, it has replicated into two. Initially we said replication is a procedure in which the double helical structure of DNA is divided into two identical double helical structures, in which both of these will have one original strand. Like if I say biologically, one template strand and one new strand. Now as it has one template strand and one new strand, both the strands are new strand. So if you see, it has one template and one new. Because of this, it is the entire DNA replication process is called as semi-conservative. Why? Why do we call it as semi-conservative? Because from the original DNA strand, whatever these two strands are there, is there in, like we got two new strands. Each strand have one original strand from the mother DNA, just naming it as mother DNA, and one new strand that is formed over here. See, this is the new one. So because of this, the half genomic constitute is conserved over here, conservation. Semi-conservative. This is called as, because it has one original strand, it is called as semi-conservative. So that is how from one strand, two DNA strands are formed with a replication process with the help of enzymes like helicase, primase, DNA polymerase, exonuclease, ligase. All these enzymes work together and help in formation of two strands. So I would like to end this demo lecture and say thank you so much.